traditional or hosts for some of these ticks, and so might
could it
they
be
have a higher tolerance of the animal?
Link
so
of
might it be an
association.
actual coevolution between the two, the three, I guess, species at this point, and so that the larger species are also less tolerant of the ticks and less tolerant.
But over
the
time might become more tolerant.
Yeah.
Yeah. We know that Lyme disease has been around for a very long time. So it's been a part of northeastern United States, for example, for centuries. So when it was sort of here and naturalized here, all of those host species were here, right?
Okay.
Those are all native host species that they would have encountered. I do think that it is likely to be a coevolutionary pattern, right? And that's what I meant by pathogens should adapt to the most common host. The host that you're encountering most often is the one on which you would co-evolve. And if you're encountering a host in habitat after habitat after habitat, there's going to be very strong selection for you to get really good at circumventing, being transmitted through that host.
Also because the immune responses are costly. And so if the mouse is downplaying some because it is always encountering the Lyme disease and the ticks, then they may just, I know that with fleas and hosts, if the fleas are on the host for a really long time, the host basically shuts down the immune response to the fleas.
An interesting point, and in fact, we're really interested in that phenomenon. We're exploring the effects of Borrelia infection and also tick infestation levels on mice in particular to try to get at that. And I'm saying this a little bit preliminarily, but the bottom line is that being infected with Borrelia or having lots of ticks on you seems to increase your lifespan and your basic demographic parameters if you're a mouse. And this is something we're really interested in exploring further.
Yeah. Because from the fleas, flea perspective, host perspective,
that would be
their immune response.
So you're saving energy.
Their port levels and their metabolic response all decreases the longer they're infected.
Right. So we're interested. We've got the phenomenon of that occurring over, tens of thousands of trapping instances in mice, but call your friends at NSF and tell them they should let us explore this more rigorously by getting inside the host a little bit as well.
So related to that, what do you think the value is of looking at variation within host species, looking at demographic variation and immunological function, life history traits, and ecological differences within species rather than interspecific?
I think the comparisons across species are critical, right? That's where we have to go. One of the things that's really challenging about doing that, though, and you should all jump in on how you feel about this, but it seems to me there's this sort of technical challenge of for example, being able to make cross-species immunological comparisons that are considered acceptable, right? We know we're making compromises if we do that, but that are considered acceptable, right? That it's good enough and we'll buy it. We validate it enough that we know what we're dealing with. I think the other thing is the problem of covariation, and that's essentially, I've just scratched at the surface of that here, that we've got this tremendous amount of covariation between density and life history and immune variation and stuff. So how do you tease apart the influence of any one of those factors? If you're just looking at comparisons, you've got all the problems of correlations, but then how are you going to get in there and make manipulations that have any kind of meaning? Marty's done some interesting work on this regard by looking at a single species, but again, within a species, where its life history strategy varies across its geographic range. Then you can start looking at, you can start breaking apart some of the phylogenetic constraints, for example, right? But it is a sense, in my view, essentially the essence of the problem. How do we get in there and make cross-species comparisons with this set of intercorrelated variables to isolate not just what is the mechanism, because I think that's probably naive, but what are the relative contributions of these different possible mechanisms? That's the million-dollar question, and I'd love to hear from other people about ideas. Yeah.
A really simple question. Why hasn't Lyme disease moved into other really small mammal mice populations extensively throughout the years? So if you go down to the southeast coast and you're looking at all sorts of other small mammal species, it's nowhere near the level.
Yeah. Yeah, and ticks are there.
And there's huge mouse populations in those regions too. And even white-footed mice go all the way down to Tennessee.
Yeah, no one knows. Gene Sau's group at Michigan State has been looking at that, trying to figure out what that, to get at the details about host communities and all of that, to get at it. I mean, we speculated that diversity is higher, actually, if you look at a very coarse scale. We speculated about this in 2000, that at a very coarse scale, diversity is higher down there. So it may be that just a cartoon of this system, right? That the pathogen, there isn't just one super great host, like the mice aren't as overly abundant as they are up in the Northeast in these sort of fragmented habitats. But it's an empirical question, right? That part, right? And you really see that. At the level of states and regions, which is the only kinds of data that we had at the time, there was a correlation with that. Sort of statewide diversity of hosts was negatively correlated with Lyme disease incidence per capita, right? No one would argue that that's a good way to ask that question. So we're really hopeful to see the results out of Jean's lab where they're doing that at site-specific areas, right? But diversity would be one possible explanation, right? Yeah, there are other ones that have been put out there, but I can't answer it. Yeah.
So a colleague of mine did a study between humans and chimps to try to get this question of the magnitude of immune response to two different species. And the way they did it was to use an antibody, a mitogenic antibody that recognized the cell surface molecule on T cells. So it was identical between the stimulatory and identical between chimps and humans. And so that took the pathogen out of the equation. And of course, his hypothesis was that humans were different on other primates and not. But that's one way of doing it, but then you have to have those kinds of reagents.
Right. So how broadly could you use that across primates?
But not down, not in the rodents.
But you could use it across primates.
Same antibody doesn't work.
Yeah. I mean, that's the trick, right? Or one of the tricks is to get something that has great taxonomic breadth. I mean, I think within primates is pretty good, right?
Within primates is pretty good. The question of the range of the bacteria, it must be the ecology of the bacteria itself, because I'm just remembering the history of Yersinia, which started in about the 14th century, just in the foothills of the Himalayas. And then with increased trafficking around the world, it spread to all rodents everywhere, including the Western Hemisphere. So, it must be that some bacteria are able to spread like this, and other bacteria just have different needs.
Right. I mean, we see Borrelia is around all those places, right? So that, I mean, Borrelia is found. If I show that map, it's at least certainly in the southeast. Borrelia is there, right? So it's getting there. The question is,
penetration is not so.
It's not. So what's preventing its deeper penetration, right? And that could be something about the nature of the host community. It could be an interaction of that with climate, right? One of the details of that graph showing the 2-year life cycle that I didn't go into is that the timing of the nymphal peak from one year and the larval peak of the next year is actually really important in this system, and it could be that in this, let me just show you that.
Strict fasting and stuff.
Yeah. This is stuff that Nick Ogden's group, who's working with Jean, is really interested in. Okay, so I showed you this 2-year part, the black line, but what I didn't show you was this. These are the nymphal ticks that emerged from the previous year. So that's this bump, right, of the nymphal ticks, but from the previous cohort, remember? And remember that those nymphal ticks are carrying the pathogen. So when these nymphal ticks, what you see right now in the Northeast is a bunch of infected nymphs out feeding. Who are they feeding on? They're feeding on the host community and infecting them anew, especially with things like mice and stuff where they're a bunch of newborn mice that have been born since the last year who are uninfected. They're getting inoculated with Borrelia right here, right, during May, June. When's the larval peak where those same hosts that are being fed on here could potentially transmit it to larvae? Right after. Does that make sense? So the relative timing of these two peaks is so important. If this peak didn't happen right here, then the host that these larvae are feeding on might not be infected. If this were over here, you could break the whole cycle. Does that make sense? And so that the seasonal timing of this may be very important. It may have to do with some of the disjunction. The relative amount of timing, the relative length of that time and position of that time may explain it. Maybe that the Northeast is sort of this perfect storm of the combination. But again, we're not sure. There are models that try to get at that and suggest that might be part of it. It's probably not all of it.
not all of them got any pathogen when they were feeding on a host.
Right.
And maybe it's just because they're feeding on hosts that don't have it.
Right.
Or,
Right.
But it seems like there was definitely a pattern when they're feeding on specific hosts, they didn't get it. Those are feeding on the night, they were more likely to get it. So.
Right. So could that just
I'm
be?
wondering if there's something.
Right, or some hosts clearing it faster than other hosts, so they're simply not infected at the time that that tick is feeding on it.
Yeah, or if there's resistance, if the tick has any resistance based upon the host.
Oh, that's interesting. I think that we don't know about. I mean, we could speculate because it's consistent with some of our data that might be true. What we can do with the this idea that the reservoir competence is combining two numbers, which is the probability that the host itself is infected, and then the probability that if it's infected, it's transmitting it to a feeding tick, right? So I showed you an agglomerated variable. But we can actually separate those two things out using maximum likelihood approaches. Jesse Brunner has taken the lead on this. And we have some just plain empirical data. Like we can ask which hosts of ours are infected. And they're basically all infected. They all have Borrelia. Okay?
There's some variation in the tick susceptibility to
Something at that tick host interface, yeah. But the hosts for the most part all have it, right? Again, partly driven by this timing, of course, that they've just gotten freshly inoculated by a whole batch of nymphal ticks before the larvae are out. Yeah.
Do you have any data on kind of the quality and the amount of the blood meals that they're getting from the different hosts, and if that ties into the question Angeline should have?
We don't have great data on that. We have a little bit. So when we put the ticks out in soil cores, we knew which host they fed on, and we weighed them all. So we knew they had fed on a host as larvae, and they came off, little blood balls,
Yeah.
and then we could weigh them and see how much they weighed before we put them out. And it looked like there was a signal, the beginnings of a signal that maybe they were having a bigger blood meal, that the hosts that had the highest, molting success,
Yeah.
the ticks on which they did the best, they had a bigger blood meal on the others, but it just didn't hold up when we looked at it
Okay.
with, controlling for things. It really didn't hold up. I thought that maybe they weren't getting as big a blood meal on opossums and squirrels, or as big a blood meal or as high quality a blood meal. Well, big would be enough, right?
Yeah.
Maybe they just can't get, those are of our hosts maybe the toughest skin, so maybe it's just, integument thickness or something, but it just didn't hold up. So what looked like maybe the beginnings of a pattern didn't hold up when we sort of controlled for things and looked more carefully at our data. I was really disappointed, actually. It was one of those results that would have been nice to have, but yeah, Pete.
I'm curious, you talked about the relationship between larvae and nymphs and sort of the lack of relationship in terms of their density between one year and the next.
Yeah.
If you know something about host diversity or host community composition, and you include that as sort of an interaction term, does that actually give you more explanatory power based on the sort of molting idea?
Yeah, when we look at the diversity, so we can, it appears that you don't need very many larvae to convert into the nymphs, right? You can see it here, right? You've got a big larval peak, right? There are a lot of larvae, and only a handful of those make it to the nymphal stage. So it's not the size of this. The argument is it's not the size of this that determines how big this is. It's the availability of hosts and the relative composition of host community that determines this. You could have this many if they were feeding on a really high quality host community and they would make it here, right? That's what our data suggests. So our diversity data with molting success and all that actually really highly predicts the density of nymphs you get out of a fixed larval community. Of course, if you had no larvae at all, they're not going to convert here. But again, the larvae appear to be able, the adults appear to be able to feed just fine on other things than deer, so you can get a larval community out of just about anything. The place where you see the role of deer unequivocally is when there really is nothing, there are no hosts but deer in a place. You've sort of extirpated mice and chipmunks and all that some way, right? And then you remove deer, then you've got nothing for ticks to feed on, and then you interrupt the cycle. Yeah. No, they don't. That is unlike most of the other tick-borne pathogens like Babesia, and Anaplasma is a big one where they do clear it, but for Borrelia they basically maintain the infection. It does seem it does decline. So if you looked at sort of titer, over time, it does decline, and it seems to since the time of the last inoculation, and it seems to decline in a somewhat species-specific way. So Mice, it won't be surprising at this point, mice keep a pretty high titer for a very long time, whereas other hosts decline more quickly, but they seem to maintain it. They don't pass it vertically. So you start over from scratch with every larval generation. And again, that's not true for all the tick-borne pathogens, but that's true for Borrelia. Yeah.
So with Lyme disease in Europe, you get similar patterns. Can you explain, are there similar species playing similar roles?
Yeah, it's more complicated. There are multiple competent reservoirs. So I can't explain the difference from that system. That's a system where the same sort of simple pattern doesn't play out. And it seems to me that it's as I said, one of the reasons we need to get at these underlying mechanisms is so we can understand when would we expect to see this kind of pattern of diversity in disease and when wouldn't we expect to see it. I mean, I think this pattern is quite prevalent, but I certainly don't expect to see it everywhere. I think that immunology might be the beginnings of a clue that we could understand something about the contributions of these species in that way, but I can't explain the differences.
Is it co-evolved in Europe? I mean,
Yeah, it's been there for quite a long time. I mean, presumably it came here from there.
Kind of building on that, it seems like a lot of the examples from your Nature paper are vector-borne diseases. Is it true that there is kind of a bias towards these patterns in vector-borne diseases? Because that sort of implies to me that immunology may not be the only, you're not saying it is the only mechanism, but that there might be an additional role for sort of
something
the vectors.
Vector feeding or something.
Right.
Yeah. I mean, again, when we start 10 years ago, when we were sort of first seeing these. Yeah, sorry. The question is, in the Nature paper, if we looked at that table and we went through the couple of dozen published papers that had shown this pattern, it looks like maybe there's an emphasis on those being vector-borne pathogens, and so would being vector-borne sort of illuminate the mechanism, whether it's immunological or something else? I guess when we were first publishing on this about 10 years ago, we thought that we actually wrote a paper where we argued that it was vectors and vectors were so important, and so we predicted it in other vector-borne disease. And Andy Dodson has argued a case for it sort of being most likely in certain kinds of pathogens, vector-borne being most prevalent among them from a sort of theoretical point of view. But the reality is the second best disease system for which this is understood is probably hantavirus. Well, West Nile now would be a competitor. Mm-hmm. And then we've got Phytophthora ramorum directly transmitted. So there are lots of directly transmitted. And in going back to the question about plants and whether it's really just density or whatever, in plant systems, this phenomenon, this is widely understood in plants, right? It's in agronomy, people understand you plant a diverse community to reduce transmission of things like fungal rusts and things like that. So direct transmission and vector-borne transmission are both happening. Whether, as we understand more about the underlying mechanisms, we can start to separate those out and fine-tune when you'd see one, when you'd see the other, and where you'd see categories, I don't know. But it's certainly happening in both, in lots of examples.